Hey there, science fans! Have you ever wondered how you grow, or how a tiny seed transforms into a giant tree? The answer lies in a fundamental process called cell division. Just like we need more people to build a bigger house, living organisms need more cells to grow and repair themselves. That's where cell division comes in. One type of cell division is called mitosis. It's like a carefully choreographed dance where one cell divides into two identical daughter cells. Imagine a cell as a busy factory. Before it can divide, it needs to make copies of all its machinery, especially its instruction manual, the DNA. Mitosis ensures that each daughter cell gets a complete set of instructions so it can function just like the parent cell. To understand how mitosis works, we're going on a microscopic adventure today. We're going to use a common onion to witness this amazing process firsthand. Ready to unlock the secrets of mitosis? Let's get started. Every good experiment starts with the right tools. Without the proper equipment, even the best planned experiment can fall short. Here's what you'll need for our onion root tip experiment, a simple yet fascinating way to observe cell division. 1. An onion. A regular onion from the grocery store will do just fine. Look for one with fresh roots starting to grow at the bottom, the fresher the better. 2. A glass jar. A small, clear jar will be our onion's temporary home. Make sure it's clean and transparent. 3 toothpicks. We'll use these to suspend the onion over water. They need to be sturdy enough to hold the onion in place. 4. Water. Plain tap water is all we need. Ensure it's enough to cover the roots but not submerge the entire onion. 5. Scissors. To carefully snip off the onion root tip. Sharp scissors will make a clean cut. 6. Microscope slides and cover slips. These are essential for viewing our specimen under the microscope. Handle them with care to avoid scratches. 7. A microscope. A compound light microscope with at least 400 times magnification will give us the best view of mitosis. Ensure it's properly calibrated. 8. A dropper, for transferring liquids precisely. Accuracy is key in scientific experiments. 9. Acetoarsine stain. This special stain helps us see the chromosomes more clearly. Handle it with care as it can stain surfaces and skin. 10. Forceps, for handling the delicate root tip. They allow for precise manipulation without damaging the specimen. 11. Paper towels, to keep our workspace clean and dry. Spills can happen, so it's best to be prepared. Make sure you have all your materials ready before you begin. Safety first. Always wear safety goggles when working with stains and sharp objects. A clean and safe workspace is crucial for successful experiments. Now comes the fun part, preparing our onion root tip. Follow these steps carefully and you'll be on your way to observing cell division in no time. First, growing the roots. Fill your glass jar with water, leaving about an inch of space at the top. This will provide the necessary environment for the roots to grow. Carefully insert 3 toothpicks around the middle of the onion, spacing them evenly. These toothpicks will help suspend the onion over the jar. Place the onion on top of the jar, making sure the bottom touches the water. This ensures that the roots will have access to the water they need to grow. Keep the onion in a dark place for 2 to 3 days to encourage root growth. Darkness stimulates the roots to grow longer and stronger. Second, snipping the tip. After a few days you'll notice white, thread-like roots growing from the bottom of the onion. These roots are what we need for our experiment. Using scissors, carefully snip off the last 1 centimeter of a root tip. This is the part where the cells are actively dividing, which is crucial for our study. This is the region where cells are actively dividing, making it the perfect sample for observing mitosis. Third, softening the tip. Place the root tip in a watch glass and add a few drops of 1 molar hydrochloric acid. This step is essential for breaking down the cell walls. This helps break down the cell walls and makes it easier to spread the cells on the slide later. Proper softening is key to a successful slide preparation. Leave it for 5 minutes. This waiting period allows the acid to work its magic on the root tip. Fourth, rinsing and staining. Carefully transfer the root tip to a clean watch glass using forceps. Handle it gently to avoid damaging the delicate root cells. Rinse it gently with water to remove the acid. This step is crucial to ensure that the root tip is clean and ready for staining. Now, add a few drops of acetoarsine stain to the root tip and leave it for 10 minutes. This stain will highlight the chromosomes, making them easier to see. This stain will bind to the chromosomes, making them visible under the microscope. The vibrant color will help you identify the different stages of mitosis. With our onion root tip prepped and ready, we're one step closer to witnessing the magic of mitosis. Get ready to observe the fascinating process of cell division. Now that our onion root tip is stained and ready, it's time to prepare the microscope slide. This slide will act as our window into the hidden world of cells. First, placing the tip. Using forceps, carefully pick up the stained root tip and place it in the center of a clean microscope slide. Make sure the root tip lies flat on the slide. Second, adding a cover slip. Gently lower a cover slip over the root tip. Avoid trapping any air bubbles, as they can interfere with our view. Third, squashing gently. To spread the cells into a thin, single layer, carefully place a folded paper towel over the cover slip. Press down firmly but gently on the paper towel with your thumb. This will squash the root tip and make the cells easier to see. Our microscope slide is now prepped and ready for its grand debut under the microscope. Section 5. Focusing in, using your microscope. The microscope is our key to unlocking the secrets of mitosis, allowing us to see the process of cell division in stunning detail. Here's how we'll use it to observe the different stages of cell division. First, positioning the slide. Place the prepared slide on the stage of your microscope and secure it with the stage clips. Make sure the slide is flat and stable. Next, starting low. Begin by focusing the microscope on the lowest magnification, usually 40 times. This helps you get an overview of the slide. Use the coarse adjustment knob to bring the image into focus. Turn it slowly to avoid overshooting the focus point. Then, centering the root tip. Use the stage control knobs to move the slide until the root tip is centered in your field of view. This ensures you are looking at the right part of the slide. After that, increasing magnification. Slowly switch to a higher magnification, 100 or 400 times, to get a closer look at the cells. Be gentle to avoid damaging the slide. Use the fine adjustment knob to sharpen the image. This will help you see the details more clearly. Finally, exploring the slide. Move the slide around to observe different areas of the root tip. This allows you to see the variation in cell division across the sample. Look for cells that are in various stages of division. Each stage has unique characteristics that are fascinating to observe. They will appear as small, darkly stained structures within the cells. These structures are the chromosomes, which are crucial for cell division. Get ready to be amazed by the intricate world of cells magnified right before your eyes. The complexity and beauty of cellular structures are truly a sight to behold. Section 6. Prophase, the cell gears up. As we peer into the microscope, we'll see cells in different stages of mitosis. The first stage, prophase, is like the warm-up act before a big performance. During prophase, the long, thin strands of DNA inside the nucleus begin to coil up tightly. This coiling condenses the DNA into visible structures called chromosomes. Imagine taking a long piece of thread and winding it around your finger. It becomes shorter and thicker, just like chromosomes during prophase. As the chromosomes condense, the nuclear envelope, which surrounds the nucleus, starts to break down. This is like removing the curtain before a stage performance, revealing the chromosomes to the rest of the cell. Prophase sets the stage for the main event of mitosis, where chromosomes are carefully divided and distributed to the daughter cells. Section 7. Metaphase and anaphase chromosomes. Take center stage, get ready for the main act. Metaphase and anaphase are when the real action happens in mitosis. During metaphase, the chromosomes line up neatly along the equator or middle of the cell. This precise alignment is crucial to ensure that each daughter cell receives a complete set of chromosomes. Imagine a perfectly straight line of dancers. That's what chromosomes look like during metaphase. Now it's time for the big split. In anaphase, the chromosomes split at their center, and the two halves, called sister chromatids, are pulled apart by spindle fibers. These fibers act like tiny ropes, pulling the sister chromatids towards opposite poles of the cell. Imagine the dancers suddenly splitting up and moving to opposite sides of the stage. That's anaphase in action. Metaphase and anaphase are like a well-rehearsed dance, making sure each daughter cell gets an equal share of genetic material. Section 8. Telophase, the grand finale. After the dramatic events of metaphase and anaphase, telophase is like the curtain call of mitosis. The separated chromosomes, now at opposite ends of the cell, begin to unwind and become less visible. It's like the dancers taking a bow and relaxing after their performance. New nuclear envelopes form around the two sets of chromosomes, creating two new nuclei within the cell. Finally, the cytoplasm, the jelly-like substance that fills the cell, divides in a process called cytokinesis. This creates two separate daughter cells, each with its own nucleus and a complete set of chromosomes. Imagine the stage curtain closing, separating the two groups of dancers into their own spaces. With telophase, the process of mitosis is complete, and we have two new cells ready to grow and carry out their functions. Section 9. Mitosis. A microscopic marvel. Congratulations, science explorers. We've just journeyed through the microscopic world of mitosis and witnessed the incredible process of cell division firsthand. By preparing an onion root tip slide and observing it under a microscope, we've gained a visual understanding of the four main stages of mitosis: prophase, metaphase, anaphase, and telophase. We've seen how chromosomes condense, align, separate, and ultimately form two new nuclei, ensuring that each daughter cell receives a complete set of genetic information. Mitosis is essential for life as we know it. It's the driving force behind growth, development, and repair in all living organisms. From a single fertilized egg to a complex human being, mitosis is the process that makes it all possible. Now it's your turn to try this experiment at home or in the classroom. Gather your materials, follow the steps, and prepare to be amazed by the wonders of mitosis right before your eyes. Don't forget to share your observations and ask questions. Stay curious, keep exploring, and never stop learning.